Well, thank you, Ronnie, for the introduction, and I'd like to thank Ronnie and also Sarah Davidson at Venega for the invitation to summarize the 2013 BGRI technical workshop. And so what we really have this year is an emphasis on the collaboration of scientists that we have from around the globe, 47 different countries. And it's appropriate that we have had this meeting in India on the 50th anniversary of Borlaug's first arrival. And I'd also like to thank Indu Sharma and the Indian Council of Agricultural Research for hosting us here in India. And this is the place where Borlaug's Green Revolution really had the biggest impact. And so the highest improvements in wheat productivity and millions of people's lives being saved in averting starvation. And so really having the meeting here in India is an amazing opportunity. And so really the emphasis here is to note the collaborations that have been achieved and also the technologies that have been produced by this collaborative community. And so Borlaug was very good at bringing technologies to the farmers. And so when Sanjay Rajaram was here, he really emphasized the point of these technologies making it to the farmers' fields, and this is really an important opportunity for these farmers to achieve that. And so I'm just going to move on and start the presentation. Is there a cue to move? Oh.
No, not that one. Okay. There you go.
And so this collaborative community is really denoted in this picture here of Indu Sharma here on the cover of Science magazine. And this shows the article that had Indu Sharma in it really showed the scientific expertise that's present here in India, and emphasizing that not only around the United States, in developing world, but also in India, and scientific expertise is really taking off. And in the second cover of Science, you see the highlights of SR33 and SR35 cloning papers that were published. And so our BGRI community has really been successful in having this biotechnological advancements, SR33, SR35 being cloned, and Evans Laguta's paper showing the cloning of LR60, SR55, LR67. And so we've made these dramatic technological advancements in the context of this broad international collaborative community. And so this is the framework to move on into the future. And so to summarize the meeting, I'm really going to emphasize the collaborations that we've seen here at the meeting. And so starting off with Dave Hodson's work, he showed how just in 2006, the wheat survey across the globe was really limited in Eastern Africa. And so with the exception of many national programs in the United States and Australia, it was just a small area that was being reported in the Rust survey. And in 2012, we have a much wider and broader survey that's being published on the Rust website. And so this is very important to know about the Rust that's out there, to have collections that can be used for research, and also to know the impact of what this community has done. And so again, in Dave Hodson and Chris Gilligan's presentation, they showed these two slides, these two pictures of Ethiopia, the severity of yellow rust, and also the presence of resistant and susceptible cultivars. And you can see in 2010, you had susceptible wheat and a high severity of yellow rust. But in 2012, as the resistant varieties moved in, yellow rust severity dropped dramatically. And so this is just showing how we can now monitor the impact of this research at this national and even global level. And so these race surveys are really key for that to happen. And building off of those surveys is the pathogen genomics work. And so here's a slide from Les Sabo showing the relatedness of different isolates of Puccinia graminis. And this is really the first stab at the global population of Puccinia graminis, showing that UG99 is very distinct from the other isolates, and it also provides the resources that we need to start going after more avirulence genes. And this topic was also covered in the poster presentation by Peter Dodds, where he has a candidate avirulence gene corresponding to the resistance gene SR22. And so this increase in pathogen genomics has been facilitated by the increased race surveys that are out there. All of this information is deposited on the websites that are available to the community. And these resources are new and they're very special, but they're really built upon the foundation of a lot of work in national programs, such as the work that Eugene talked about in the United States, the annual race surveys that have been going on for decades. The work in Australia, race surveys going on there. We're seeing more and more race surveys, race typing, survey of rust severity in the field, that's being made available. And not only that, we also have an increase in the capacity and facilities to do even more within a regional setting. And so, again, the Rust Center that's been established in Izmir, Turkey is going to play a pivotal role in race surveys in the Siwana region, and we're really excited to see an increased capacity there. And speaking of capacities for race analysis, also the ability of this community to make research progress has really been ramped up by this really critical screening facility in Njoro, Kenya. And we are really indebted to Sridhar Bhavani, formerly Devinder Singh, the leaders of CIMMYT, and the Kenyan collaborators, Peter Zhao and Ruth Wanyera, and really enabling this community to get the data that we need. And so many of the presentations, if not most or all, presented data that was gathered from this international nursery. And of course, Peter Zhao discussed how this nursery was used in his breeding program. And so this is a really key collaborative site that has facilitated a lot of our progress. Another interesting collaboration that's emerged as we've seen stem rust, we've achieved all this progress, and now the community is recognizing the really immediate threat of yellow rust. And Cristobal Wowie presented his work in the beginning of a SkipRid project in order to understand both the pathogen biology and genomics of yellow rust, but also to improve wheat resistance to yellow rust. And again, this was an international collaboration that came together and is going to provide for really exciting results into the future. And speaking of more of the results that we've seen at this meeting, we've seen a few students come and give really excellent presentations, like Paola Silva's presentation here of the effect of different minor gene QTLs and how they interact. But as you browse through the poster session, you see that the really great number of resistance genes, QTL, a lot of work that's really the backbone of what we do. And a lot of that wasn't highlighted in the oral presentations, but it's really the foundation of why we're here and what the products are that we're delivering. And also the Ethiopian graduate student who presented his work on the identification of significant QTL that are associated with resistance in synthetic wheat. And so you've seen not only here at the meeting and in Ethiopia, a lot of work being done to find new resistance. And in the wild progenitor species, in this work done by Dr. Chinega, she showed the resources that are out there for us to tap. And so we found a number of genes in QTLs, but there are still a vast amount of resources out there for us to tap into. And so it's very exciting to see the progress that she reported there. So Renee Prins and also Susanna Dreisigacker presented their collaborative research project and also on both association mapping and fine mapping of both yellow and stem rust resistance. Their truly global collaborative project is producing really high impact, very relevant research. But what I wanted to point out in this slide is the gender ratio just based off of the pictures here. And so this is kind of, I think, a very good symbol of what the BGRI is kind of about and how we're kind of moving into the future. And so we've seen increasing amounts of women involved, women scientists, women graduate students, and I think this is a very good trend that this community is setting for the agricultural scientists as a whole. And also we recognize many of the graduate students and postdocs, early professionals, early scientists who are women who are moving into the agricultural career, and we support them and welcome them and recognize Dr. Yu Jin's contribution to many of these people and encouraging them in their early careers. And a really neat collaborative project that was also described in this meeting is the Pakistan project with Kay Simmons from USDA and also Dr. Mujahid in Pakistan. And this really comprehensive project included pathogen biology all the way to the distribution of a resistant variety of wheat in ARC 2011. And so you can see how this collaboration that was primarily between the US and Pakistan, but very much relied on the use of Simmet's material and to have a really great impact on the world for increasing food security. Dr. Prabhu from India reported on the vast amount of work that their group is doing in India to breed additional resistance genes into the adapted backgrounds, and these also will be genetic resources that will be useful for breeding into the future. And then when Jim Anderson was describing his breeding project, he emphasized several things which I think are very important points. And one of those is, of course, the collaboration he needs in order to get the phenotypes in Africa, but also the number of phenotypes that he gets along the whole breeding process of his breeding program. And you can see that in order to really have a good ability to breed for adult plant resistance, you need lots of phenotyping. And many of the speakers alluded that though we have a lot of new genomics tools and breeding technologies, we are really in lack of phenotyping right now, and we really recognize the importance of phenotyping. And sort of moving on into like these new technologies that are available, the representative from Mako indicated her work in showing how they have this transgenic trait for nitrogen use efficiency in rice and also in wheat. And she emphasized the public and private partnerships that are necessary and facilitate the distribution of these traits into farmers' fields. And so it's exciting to see this work being done, and also hopefully we'll get into farmers' fields soon. Interestingly, one of the very good graduate student presentations that was made by Aurora was the use of these NDVI measurements to measure stripe rust severity. And so this is an advance in phenotyping, which could be very critical. There's been some work in this type of phenotyping in the past, but really applying it to yellow rust and using it in breeding is very significant, and hopefully is a trend that will be used in the future in order to provide the more phenotypes that we need and very accurate phenotypes. Another really emerging technology that was presented here by Dr. Nirmala was the use of these RGD peptides in preventing the rust spores from germinating. And so potentially there are some options long-term in order to use this type of technology in preventing rust diseases. Jesse Poland presented his description of genotyping by sequencing and the multiple different applications that this have. And so not only in genotyping material, genetic mapping, association mapping, even use of the markers in the spike GBS. And so as these technologies are becoming cheaper and cheaper, it's going to be much easier to use them in breeding and across the world because of their lower costs. And then Kevin Smith presented his work in genomic selection, and it's really exciting to see that in a trait that is as difficult as Fusarium head blight, they've made very good return from selection, from genomic selection for this trait of low amounts of DON. And so this is very encouraging that genomic selection could be used for very complex traits such as yield. And we're looking forward to seeing the results from Jessica Rutkowski and others who are working on genomic selection in rusts. And so there's several key things that emerged from this meeting where speakers came to the floor and really called for increased participation of our group in order to facilitate further collaborations. And so one of those calls to action really was made by Chris Gilligan from Cambridge University. And he and Dave Hudson are working on developing these risk and hazard maps, and they really needed additional information on the environmental sensitivities of the pathogen and the dispersal capabilities of the pathogen within fields. And so this is really a call to the community as a whole, and I'm just reminding you that if you have such data or are able to generate such data easily, to contact Chris Gilligan or Dave Hudson to try and integrate this into their work to generate these risk and hazard maps, which could be very useful in mitigating risks in the future. Another call to action was made by Les Sabo, and in their work with the Puccinia graminis genotyping and understanding the world distribution and genetic diversity of Puccinia graminis, they don't have a very a widespread, even representation of Puccinia graminis isolates. And you can see many countries they do not have any isolates from. And so this is, again, a call to action to many of you in this room to increase your collaboration specifically with Les Sabo and Botma Vissa and their project. And I think the results from this project are going to be very useful to wheat breeding as a whole, to know about the distribution of a virulence effectors in the global population. And this is going to be something that we can do right now to really facilitate the long-term durability of resistance. Another call to action was made by Bob Bowden, and there was a lot of discussion about gene stewardship. And a lot of points were raised, a lot of debates and disagreements. But I think we have a general consensus that we do need to actively steward our genes. And the question we need to address right now is how to effectively do this. And what was called for by Bob is to join in participation in discussing what the appropriate methods for stewarding the genes are. And so one way you can do that is to join the listserv that is actively discussing these topics. And hopefully in the near future, within the next few years, we can have a consensus of our strategy in order to use the major genes wisely and not be here 30 years from now discussing the same exact issues. Bob Bowden also discussed pyramiding genes specifically and combining multiple genes, the exponential ability of combining 5 instead of 3 genes and such. And so I'd encourage all of the breeders especially to provide their inputs into this process and to be diligent in deploying genes effectively. It only takes one breeder to kind of destroy a pyramid if these genes are separated individually. And so to kind of move on from the different collaborations and technologies that we've observed, and going off of Borlaug's legacy of taking it to the farmer, and I think that there's a number of things that we can do to take it to the farmer to sort of finish this project off. And as many of the symposium discussed, there are some options on the table that have been suggested. And one of those is to figure out what we have to do next as a global community. We have this great catalytic ability as this really close collaborative community to have high productivity, but we're really dealing with a very small part of the yield gap, as was demonstrated by Iago Hale in his presentation. So the question of the future is, how are we going to address the other limitations in yield, and how are we going to collaborate or invite other groups in to really take it to the farmer and close this yield gap? This is going to take a lot of strategic effort, and I think this is going to be where we can be very productive in helping people out. This community is very collaborative and very close, and I think it's a very good model for the entire research community as a whole. And lastly, when Ricken was here and discussed digital green and the amazing extension system that is available there, it was really amazing to see the impact that that has. And so moving into the future, definitely the BGRI community should seek to integrate with extension systems that are more relevant for today and more effective. And so such collaborations and growing our community will really be effective in taking it to the farmer. And I'd just like to close by recognizing Dr. Borlaug's legacy in India, the farmers who were here and their testimonies of their reverence for Dr. Borlaug, their deep respect and thankfulness really sets the bar high. And I hope that we can continue collaborating to bring our technologies and take it to the farmer. So thank you.